Hi, my name is Zach Hettinger, and I, along with colleagues at Purdue University, am delighted to share our recent work out in October's issue of Experimental Physiology. One hallmark feature of aging is a phenomenon described as cellular senescence, which is the irreversible proliferative arrest of the body's stem cells, ultimately leading to tissue decline and poor regenerative capacities. In addition to cell cycle arrest, senescence is also accompanied by a pro-inflammatory senescence-inducing paracrine effect on neighboring cell populations through what has been characterized the senescence-associated secretory phenotype, or the SASP. Skeletal muscle possesses its own resident stem cell population, termed satellite cells, which are responsible for tissue regeneration and crosstalk with both muscle fibers and resident stromal cell populations. The non-fusion effects of satellite cells are becoming increasingly realized, in part through the release of small extracellular signaling molecules termed exosomes, or EVs. While the effects of cellular senescence and proliferative arrest have been reported in muscle stem cells, the effects of the SASP on satellite cell crosstalk, particularly through EV communication, has not yet been reported. A common feature of aging skeletal muscle is not only a loss of muscle mass, but also a reduction in muscle capillarization, leading to poor muscle function and repair. Capillarization is carried out in part by endothelial cells, which lie in close proximity to the muscle stem cell niche. In this study, we hypothesized that the induction of senescence in muscle stem cells results in altered EV signaling, ultimately causing the release of senescence-inducing EVs to endothelial cells in an in vitro cold culture model. Using muscle stem cells isolated and purified from the vastus lateralis muscle of young, healthy human adults, we induced premature stress-induced cellular senescence through transient administration of hydrogen peroxide. Following treatment, we observed increases in numerous senescent markers, including senescence-associated beta-galactosidase activity, cyclin-dependent kinase inhibitor P21 gene and protein levels, and a reduction and proliferation via EDU incorporation. In addition, we also observed a nearly 5-fold rise in small EVs termed exosomes in treated muscle stem cells compared to untreated controls. Together, our results show that the acquisition of a premature senescent state is accompanied by robust activation of EV signaling.
Hello, my name is Chris, and I will take you through the rest of our findings. Following EV characterization, we took human umbilical vein endothelial cells and treated them with control and senescent myoblast-derived EVs to determine if premature senescence alters the signaling potential of EVs. We were interested in how the myoblast EVs would affect endothelial cells because of the signaling axis that exists between the two cell types. We found that the premature senescent-derived EVs decreased in vitro measures of angiogenesis like proliferation, migration, and the ability to form tube-like structures. Interestingly, we also found that the senescent-derived EVs transferred a senescent phenotype to endothelial cells. This was potentially due to the transfer of SASP factors like TGF-beta. Overall, our data provides in vitro evidence that muscle stem cell extracellular vesicles could potentially contribute to impaired angiogenic and microvascular function seen in numerous senescent-associated disorders like aging, obesity, and diabetes. Thank you everyone for watching, and check out the article if you haven't already.